Cultural characteristics and colony morphology of bacteria. When grown on a medium, bacteria form colonies. Colony morphology is a description of the appearance of the colonies. Each bacteria have typical appearances or morphology. However, colony morphology does not identify an organism. It can be used to distinguish a mixed culture from pure culture. Characterization of bacterial culture in agar plates.
The factors used to characterize a culture in agar plate are as follows: pigmentation, surface characteristics, shape of colony, size of colony, margin of colonies, and elevation of colonies. Note that other factors such as odor, diameter, and pigment to surrounding medium can be considered. Pigmentation are commonly described as either black, brown, light yellow, or milky, opaque or transparent, orange, red, or pink, and white. Surface characteristics include smooth, glistening, rough, wrinkled, dull, and others, such as concentric, radiate, contoured, and mucoid. Shape of colony are usually described as punctiform, circular, filamentous, irregular, rhizoid, and spindle.
Sizes can be defined as pinpoint, pinhead, or matchhead.
Margin may either be curled, entire, or smooth, filamentous, undulate, lobate, or arrows. Elevate can be described as raised, convex, flat, umbonate, crateriform, and pulvinate, as shown in this picture for reference.
Let us use this pure culture for characterization. Its overall form is circular. Its elevation is either raised or convex, as appropriate in this case. The margin is entire throughout because its edge or borders are circle. Its optical property is opaque. Its texture or surface is smooth or glistening when an inoculating loop is used. And its pigment is a yellow color. Size, however, is varying. Characterization of bacterial culture in agar slant. Form may be defined as filiform, acuminate, beaded, effuse, arborescent, rhizoid, and/or spreading. Abundance of growth can be listed as no growth, slight, moderate, and large. Consistency of growth is either dry, buttery, mucoid, and brittle. The common pigmentations listed are green, red, and yellow. Optical characteristics are either opaque, translucent, or transparent. Liquefaction can also be considered, defined as crateriform, napiform, infundibule, sussate, and stratiform. Now let us take this sample in an agar slant for characterization. The amount of growth would be moderate to abundant and irregular. Sediment, as stated, is also flocculent. Its form is irregular, which in this case should be arborescent. With this listed characteristics, we can hypothesize the probable bacteria belongs to the genus Lactobacillus. Further characterization should identify it. Characterization of bacterial culture in nutrient broth.
Surface growth may either be flocculent, ring, pellicle, or membranous. Subsurface growth, if present, should be defined as turbid, flocculent, flaky, or granular. Amount of growth can be listed as none, scanty, moderate, or abundant. Sediment, if present, can be described as granular, flocculent, flaky, or viscid. For pigmentation of the bacterial culture in nutrient broth, it is done the same way as we define agar plates.
Let us now characterize the sample. Note that the broth was swirled to show that it is flocculent. With that said, its surface and subsurface growth are both flocculent. The amount of growth should be moderate to abundant. Sediment is also flocculent, and its pigmentation is whitish.